Hi, everyone. I'm Mariska Goswami, as Alyssa just introduced me, and I go to Troy High, and I've been working in Dr. Zhang's lab for the past 8 weeks on this project, which is identifying binding partners of the human DNA Li1 protein by yeast Dihybrid screen. So to understand the DNA Li1 protein and its function, we can back up a little bit and review spermatogenesis. Spermatogenesis has 3 major steps, which are mitosis, meiosis, and spermiogenesis. And during this process, sperm cells undergo a lot of really important changes, including acrosome and tail formation, which is important because if sperm cells cannot grow tails and they cannot move, then they cannot fertilize the egg. Before I arrived at the lab, Dr. Zhang's lab had already found that the Parkin co-regulated gene protein, which is PACRG, and mouse meiosis expressed gene 1 protein, which is MEG1, form a complex that transport cargo important in sperm tail formation. And the complex is listed here. They also found, using the same technique that we did for this experiment, that PACRG interacts with DNA Li1, the focus protein of this project, which stands for dynein axonal light intermediate chain 1. Because they found that PACRG interacts with DNA Li1, they decided to generate DNA Li1 conditional knockout mice along with mice that have PACRG and MEG1 knocked out. Overall, the phenotypes were similar, except for a couple key differences in the DNA Li1 mutant mice that were not seen in the MEG1 PACRG mice. As you can see here in this photo, the 2 sperm cells have actually not separated and they have multiple tails as opposed to 1. And because this knockout caused such a difference in the phenotype, they hypothesized that DNA Li1 might have other functions besides the ones on the MEG1 PACRG pathway. Sorry, technical difficulties. I believe it's working. Thank you, Alyssa. So yeah, I know. So from that, we decided that our goal was to explore potential new functions of DNA Li1 in sperm formation. In order to do this, we decided to perform a dihybrid library screening using yeast mating. And the way this works is it's a bait and prey situation. So our bait protein is DNA Li1 cloned in this vector, and our prey is a protein library that we've gotten from a company that's constructed in another vector. The vector that contains DNA Li1 is kanamycin resistant, and kanamycin is an antibiotic. And the protein library containing plasmid is ampicillin resistant, which is a different antibiotic. So if you tried putting the DNA Li1 plasmid in bacteria on an ampicillin plate, it would not grow. And same with the protein library and kanamycin. However, if we mated them and plated them on an ampicillin plate, then you would see growth if they mated correctly because they confer both resistance to both antibiotics. In order to do this, the steps are to mate the bait and the prey, plate the yeast on the medium, isolate the DNA from that, and transform it to bacteria. And if it grows, then we know that they mated. So that's the screening process that occurs. More technical difficulties.
You have to click back.
Thank you, Alyssa, again. So before that happens, there are a lot of preliminary steps that go into before we mate the yeast. First, before I joined the lab, Dr. Zhang's lab actually designed the primer that amplifies the human DNA Li1 cDNA, and they performed PCR with it. That's the leftmost picture. The second well only, the first well was for a different experiment, but you can see that PCR happened correctly because the correct band links are there. And then they extracted that from the gel and performed TA cloning with it, and then sent it in for sequencing. So some of the inserts that they sent in for sequencing actually had no mutations, but I wanted to point out this one because it has one mutation, but is on the 3rd base pair of the amino acid that it codes for. And sometimes amino acids, 3 base pairs code for the same amino acid, even if the 3rd pair is different. So this mutation wouldn't have mattered. and we could have still used the sequence, but in the end, we decided to go with the ones that did not have mutations. After that, when I joined the lab, we performed subcloning with the confirmed DNA Li1 insert and our vector, the one that confers kanamycin resistance. And we transformed that to bacteria and then isolated the plasmid from it and ran this gel. In lane 1, you can see that the correct insert is not there, but in lanes 2 through 6, the correct insert is seen relative to the latter. We decided to use plasmids 3 and 4 for the experiment. We also had to test for autoactivation because the way the screening process works, if the yeast grows without being mated, then we will never know whether it has mated or whether it grows on its own. So the top plate is grown on SD-3 medium, which means that it doesn't contain 3 of the essential amino acids that yeast needs to grow. And if we plate the yeast with the plasmid on this medium, but not mated with the protein library plasmid, and it grows, then we know we can't use this for screening because it grew without mating. However, as you can see on the top 2 plates, no growth has occurred. To grow the yeast before we can mate it, we use SD minus tryptophan, which is just 1 amino acid that yeast need to grow. And tryptophan is synthesized by the plasmid that we transformed onto the yeast. But the AH109 yeast strain that we used cannot synthesize tryptophan by itself. So in order for it to grow, you can see that on the bottom left and bottom right, plasmids 3 and 4, there are growth on those plates, but not in the bottom center plate because it has not been transformed with the plasmid and therefore cannot grow on a medium that does not have tryptophan. Once we grow our yeast, we need to perform a Western blot in order to see whether our protein is being expressed. We used an antibody for a tag protein that is on the same plasmid as DNA Li1 called c-Myc, and the molecular weight of DNA Li1 plus the tag protein plus a little bit of the rest of the plasmid is about 47 kilodaltons. So we would expect to see a band right underneath 50, which we do see in all of our samples, but not in the negative control. If we had seen it in the negative control, it would have been possibly a non-specific protein, and we wouldn't have known whether our protein was being expressed. So with that confirmed, we could move on to mating and plating. In order to mate the yeast with the protein library strain, we need to grow it to an optical density of 0.8. Now, up until yesterday, the yeast had not grown past an optical density of 0.15. So this is where it gets a bit complicated. I took this picture yesterday at noon and right before I sent my presentation to Alyssa, and the yeast is now growing. Now it's mating as we speak in the lab upstairs, but because of these complications with growing the yeast, we couldn't finish the project before the Discovery to Cure program ended. So I will walk through the rest of the steps for the sake of showing what we hope will happen, but the rest of this is speculation as of right now. So we grew the yeast, we will grow the yeast, and right now it's mating. What happens is we take the yeast that has grown and we mate it with the protein library strain, which is very concentrated. We don't need to grow that. And then we shake it in liquid medium for 24 hours. Then we put a little sample of it under a microscope to check for a zygote, and a zygote will be seen when the yeast has mated. So this is an example picture. And once we see that mating has happened, then we plate on the plates, the SD-3 medium that I was talking about earlier, that do not contain 3 of the essential amino acids, but we're also plating with this X-alpha-gal indicator. And what this indicator does is when the 2 yeast strains mate, they release this compound that turns the indicator blue. So the protein library strain can grow on its own on this SD-3 plate, but we don't want to just pick that up. We want to pick up the colonies that have mated. So we see that the blue colonies are the ones that we have to pick up. We'll pick up those, we'll isolate the yeast DNA, and we'll transform to E. coli, sorry, on an ampicillin plate. Then we isolate the plasmid DNA from that bacteria and send it in for sequencing using the same website that we sent the inserts in for sequencing in the beginning. Then we compare our insert, the confirmed one online, the sequence of DNA LI1, with every single plasmid that we've isolated. And we compare it against all of these, and we see how many times has the insert, has there been a match? How many times has it shown up? And from there, we count how many times it's interacted with each protein. And if it interacts more times, and it's more likely that DNA LI1 binds to that protein for a function rather than a random or non-specific binding. Possible applications of this. Before I joined the lab, like I said, Dr. Zhang had already conducted a yeast-2 hybrid screen, the same technique, but with PACRG, and found that DNA LI1 binds to PACRG for a purpose, which is they form a sperm flagella complex. Blocking this potentially would mean blocking the development of sperm flagella, and like I had said earlier, those are crucial for fertility, so it could lead to the development of male contraceptives. By that line of reasoning, if we find a protein in this yeast-2 hybrid screen that we know the function of, and we block that binding, we can predict what would happen, what it would do to the function, what it would stop. We don't know because we haven't finished the experiment, but we predict that it might have to do with sperm individualization during spermiogenesis, because the multi-tail phenotype that we saw when we knocked out DNA LI1 in mice showed sperm that had not been able to separate and had grown multiple tails. So we think that it could have something to do with that, and we're not sure. It could also serve as a new genomic marker for male infertility, because if we know that these two proteins form a complex and that they're crucial to fertility, then we can observe why is this not happening? This will be a new reason or a new explanation for male infertility. With all of that said, we don't know yet, and it was really sad to not be able to finish the experiment before the Discovery to Cure program ended, but I am really, really grateful for the time I did get to spend in the lab and the time I got to spend learning about this project and the other project that Dr. Zhang has been working on, because he was kind enough to let me work on those as well. And with that, I conclude my presentation. I would like to thank Dr. Zhang, Dr. Lee, and Candace for all of their help this summer. Sanjana and I followed them around everywhere, and we never let them take a break from us, but they handled it the whole summer. And also to Dr. Moore and Alyssa for organizing this program, because I had such a great summer. And thank you.